Om Swastiastu, good morning. We are from SGD B2, the first semester of PSSKPD 2021. Today, we would like to present our student project presentation, which title is Cell Junction in Tissue Morphogenesis. Before we start, I would like to introduce myself. My name is David Christopher Tjandra, and my name is 21025110066. I am the leader of this student project presentation of Biomedic 1. And now, I would like to show our SGD B2 members that will do this presentation. Here are the members. Now we will move on to the introduction. The human body is composed of organ systems with various organs, tissues, and cells. These elements have various forms and patterns that support their main function in doing our daily activities. Behind the various elements that exist, it turns out that many of our body elements have very unique and specific structures and patterns in shape. Some of the examples are distinctive bone shapes, patterns arrangement of mesenchymal cells, the shape of the heart with its structured blood vessels, and so much more. These structures can be formed due to a process in the body called tissue morphogenesis. Tissue morphogenesis is the process of collaborating tissues in the body to form a new pattern that becomes the main identity of an element. This process holds a very crucial and important role in the human body. During the process, the application of tissue morphogenesis is closely related to the collection of cells that are combined to form the tissue. The merging of all cells in the body is certainly not separated from cell junctions. Cell junction itself is a cellular structure that bridges communication links between one cell and another. Through it, the various cells in the body will be interconnected and coordinated together to create a strong and a good connection that will make up tissues.
Good morning. My name is Yovilia Tio Yunus. My last 3 digits of student number is 035. Now I will explain about problem formulations. The first one, what is the general interpretation of cell junction and tissue morphogenesis? Number 2, why does cell junction in tissue morphogenesis occur? Number 3, what is the role of cell junction in tissue morphogenesis in our body? Number 4, what is the disease disorder of cell junction in tissue morphogenesis? And the last one, what treatments can be done to treat the disease disorder of cell junction in tissue morphogenesis? Objectives. The first one is to find out the general interpretation about cell junction and tissue morphogenesis. Number 2 is to find out why cell junction in tissue morphogenesis occurs. Number 3, to find out the role of cell junction in tissue morphogenesis in our body. Number 4, to find out the disease disorder of cell junction in tissue morphogenesis. And the last one is to find out treatments that can be done to treat the disease disorder of cell junction in tissue morphogenesis. Benefits. The first one is for the public community. Introduce the public about cell junction in tissue morphogenesis. Inform the public about the disease disorder in tissue morphogenesis and its treatment. And increase the public knowledge about cell junction in tissue morphogenesis in their body. Next is for the institution. Adding sources of knowledge about cell junction in tissue morphogenesis as a learning material to introduce students about cell junction in tissue morphogenesis and improve the quality of learning material about cell junction in tissue morphogenesis. And the last one is for the writers. This paper can be used for medical resources, improving literacy skills by searching through a lot of medical resources and practice gathering information from various resources. That's all from me. Thank you.
Good morning. My name is Gusti Agung Ayu Saraswati. My last 3rd digit of student number is 056. Now I will explain about definition of cell junction. Cell junction is membrane site plasma that ties cells to other cells or extracellular substance. There are 3 kinds of cell binding. Number 1, tight junction. Number 2, gap junction. And number 3, desmosomes. Tight junction component lipid from 2 membrane plasma will be connected tightly bound together by membrane protein locked under tight intersection. Tight junction by effectively sequester the contact lumen from the bilateral surface of the cells. For example, tight junction near the apical surfaces of cells lining the digestive tract help keep enzyme, acid, and product was kept under control. In the gap junction as shown in figure 2.1c, 2 cells are bound by 2 interlocking transmembrane protein called connection. Thank you.
Good morning, I'm Amanda Devasya. My last 3 digits of student number is 068. Now I will talk about cell junction rule. First, cell junction is a barrier. In this rule, a cooling junction provides a barrier through the ceiling of one or several cell layers, which prevents the passage of water and some solids. The interaction of the transmembrane protein claudin and occludin in each cell causes the separation of the membranes. This sort of intercellular seal proves that chemical crossing an epithelium in both directions. In this role, cell junction also serve a related purpose in epithelia. Second, cell junction is an adhesive. Cell junctions are attached to components of extracellular matrix and anchor to each other, thus playing an important role in keeping the cell together in the structural cohesion of tissues. A desmosome or macula adherent is a related adherent junction. This connection resembles a single spot weld and doesn't form a belt around the cell, as the name implies. Third, cell junction is a communication between cells. Cell junction role is to change the pathway of chemical and electrical signal from one interacting cell to another. Connexins are the protein that create hexameric complex term connexons, each with a central hydrophilic hole. Connexin is the adjacent cell membrane laterally and aligned to form connection between the two cells when two cells attach, with each junction having dozens and hundreds of aligned connection pairs. Thank you. Next.
Good morning, my name is Edgar and my last 3 digits of student number is 045. When cells come close to each other, they develop a specialized intercellular junction that helps cells stay connected to each other, communicate, and so on. We're going to focus on 3 intercellular junctions: tight junction, desmosomes, and gap junction. The first, tight junction, or also known as zonal occludence, is a protein that becomes the tight seals between cells. Therefore, it glues one cell to another and controls the movement of molecules between them, preventing from flowing freely. Desmosomes or macula adherens is derived from the Greek words demos, which mean connection, and soma, which mean body. Therefore, desmosomes are a connection of cells with their neighboring polarized epithelial cells. Desmosomes anchor each cell through membrane to each cell's cytoskeleton. By anchoring each cell's intermediate filament cytoskeleton to its plasma membrane, providing a very strong adhesive junction between cells. And last, gap junction, sometimes referred to as communicating junction or macula communicans, are a type of cell junction that uses intercellular protein channels to connect neighboring cells. The gap junction is the place where intercellular communication occurs because it allows ions, amino acids, nucleotides, and other water-soluble molecules to move cytoplasmically. That's all for me. Thank you for listening.
Good morning, I'm Komang Ayu Widhiani. My last 3 digits of student number is 044. I'll talk about tissue morphogenesis. According to the Big Indonesian Dictionary, morphogenesis is the process of organ or tissue development and differentiation that occur in accordance with what the organ or tissue does. Morphogenesis is the biological process by which embryonic cells develop, move, and organize themselves into structures, organ, and system of an adult organism by being induced by a combination of chemical and mechanical factors. In morphogenesis, there is a collection of signaling molecules called morphogenes. The arrangement of the same specialized cell in the body that unite in carrying out certain biological function is called a tissue. Tissue morphogenesis is also necessary for the basic organization of the embryo and organs. The developing tissue can undergo a diverse series of changes such as bending, elongation, narrowing, branching, and folding. But at all confirm that embryonic tissue can control tissue morphogenesis by altering the balance of cellular mechanical forces. The complex process of tissue morphogenesis requires coordination and cortical tension between cells or tissues. Liquid confirmed that intercellular coordination has the possibility to involve mechanical pairing of cell contractility between adjacent cells. The change in shape is supported by an intercellular contractile network consisting of actin filaments, actin cross-linkers, and myosin motors. It generates the cell surface precisely in the cell cortex and affect the ability of the cell to change its shape and collectively form a shape during tissue morphogenesis. Thank you.
Good morning, my name is Nyoman Medhira Amanda. My last 3 digits of study number is 070. Now, I will talk about the basic principles of tissue morphogenesis. Morphogenesis are these atoms that act as signal molecules which can really influence the behavior of the cell. Morphogenesis in embryo repair is characterized by extensive cell development, cellular modification. There are 2 basic types in the embryo, namely epithelial cell and mesenchymal cell. Different cellular forms that happen inside the epithelial and mesenchymal courses of actions direct the method of morphogenesis. Changes in cell shape are required for morphogenesis. Epithelial to mesenchymal moves can be where epithelial cells lose their attachments to their neighbors and they end up mesenchymal. Cell growth or change to cell size. While the sperm eliminates, most of the cytoplasm becomes smaller. In addition to cell divisions and cell growth, apoptosis or cell death is also an essential part of creating form. That's all from me. Thank you.
Good morning, I'm Nathania Susanto. My last 3 digits of student number is 051. Now, I'll talk about the introduction of cell junction in tissue morphogenesis. Tissue morphogenesis is the biological process of forming new patterns using tissues to create innovative structures in the human body. A tissue is defined as a myriad of cells with the same structures and characteristics that combine together to work as a single entity. Cell junctions are mostly found in epithelial tissues. Therefore, cell junctions are definitely present and most certainly play a role in the process of tissue morphogenesis. As an example, in 2005, Liquid states that morphogenesis in the epithelial tissue is done to shape human organs during their embryonic stages, and that it relies on the shape of the cells and junctions that are responsible in forming said tissues. The shape, movement, and interaction between the cells that form epithelial tissues are determined by cell adhesion, with cell junction being one of the many examples of this. During morphogenesis, the cells that form epithelial tissues will undergo intercalation, which is the process of changing the positions of cell and its junctions. This allows the remodeling of the tissue structures. The cell junctions are removed in the beginning of the process so the cells can produce new shapes such as tetrads and rosettes, as you can see in the picture in the slide. After moving, new junctions are placed between the repositioned cells, creating a new shape of tissue. In short, cell junctions play quite a large part in the morphogenesis of epithelial tissues. The repositioning of cell junctions after finishing the intercalation process allows the tissue to maintain its new shape and structure for the body to utilize later on. That is all for me. Thank you.
Good morning, I'm Janice Wijaya. My last 3 digits of student number is 037. Now let me continue the presentation with the importance and its role in our life or body. The dynamic regulation of cell shape is essential for living organisms and also has impact on the internal organization and with the environment. Changes in cell shape are associated with migration, division, proliferation, adhesion, and differentiation within tissues. Cell shape changes are induced by internal and external forces generated from the cytoskeleton and from the surrounding cells. Morphogenesis in tissue will occur when the cells divide apoptosis forced by the environment into the epithelium. Cells in epithelial tissue undergo shape changes during development, which are drivers of both embryonic tissue morphogenesis and adult tissue morphogenesis. Epithelial cell junctions are required in the process of tissue morphogenesis. Adhesion between epithelial tissue cells is very important for tissue integrity, cell-cell communication, for epithelial and endothelial cell lining tissues, and for tissue morphogenesis. Another role of junction is formation 2 that is used for motor activity. And epithelial tissue morphogenesis is also very important for embryonic development and also have an important role for building a complex body structures and organs. That's all for me. Thank you.
Good morning, my name is Marta, Vitti, Lawi, and my last 3 digits of student number is 064. Now, I'll talk about disease disorder of cell junction in tissue morphogenesis. First, on tight junction. Dysfunction of tight junctions functions can result in various pathological conditions like Crohn's disease and renal epithelial disorder. A. Crohn's disease. Crohn's disease is a type of the inflammatory bowel disease. This disease is mainly related to the dysfunction of the epithelial barrier, which also refers to the disruption of the tight junction. With it, the intestinal epithelial tissue morphogenesis is deformed and will cause an increasing cellulose loss in the epithelial tissue. This cellulose loss resulted in intestinal barrier tissue disintegrity and increased the intestinal permeability. B. Renal epithelial disorder. Proper tight junction assembly is required during renal development and morphogenesis, but the segmentation of the renal may lead to the formation of unique nephron segments in the epithelial tissue. Another various factors also could be resulted in tight junction disassembly, increase of cells apoptosis, and tubular cell detachment. Second, on desmosome. Desmosomes have scatterings which for some people could trigger the immune system and cause a blister-like skin disease called pemphigus. This pemphigus autoimmune reaction may cause dysfunction in desmosome that leads to dysfunction in skin tissue morphogenesis that can be resulted to an abnormal morphology of the skin tissue itself. Third, on gap junction. Gap junction allows traffic of cytosol to another cells around it. Usually, the gap disorders in gap junction are caused by the mutation of connexin that could cause a rare hereditary skin disorder such as Vohwinkel syndrome, hypotrichosis Daphne syndrome, and erythrokeratoderma variabilis. So that's all from me. Thank you.
Good morning, my name is Fernando Ghosali, and my last 3 digits of student number is 036. Now I'll explain about the treatment analysis for those disease disorder. The first one is tight junction, the Crohn's disease. There is no standard therapy and cure for this disease. The best effort to this disease therapy is just to press down the abnormal inflammatory symptoms, which also let the intestinal tissue heal inside. But maintaining the tight junction activity is also important. If the junction works normally, then the symptoms also could be decreased. The renal epithelial disorder. The point of this disorder is to recover and to re-stabilize the function of tight junction. This tight junction recovery also has an impact on the recovery of the functional epithelial because the normal tight junction function holds an important role in numerous integrated processes. The second one is desmosome. The pemphigus disease treatment result cannot give a complete recovery. The treatment is by reducing the lesion improvement and growth and also prevents the recurrences of the treatment. This already can count as a successful treatment. Commonly, corticosteroid is taken as pemphigus medication, but the complication can be caused by corticosteroid. The third one is gap junction. We know that connexin is one of the gap junction protein subunits that hold the function of building up intercellular channel that present in every gap junction. But this connection is not only taking part in normal function. In some cases, this connection has a large number of mutations that can lead into skin disease. These mutations have caused dysregulation of hemichannel activity. In this case, composing the connection inhibitor and hemichannel inhibitor could be used as strategies in the clinical therapy practice that has the therapeutic valve. Thank you.
Good morning, I'm Ledwin Meikel Wibisono. My last 3 digits of student number is 061. Now I'll talk about our discussion. Based from the analysis that we have done about cell junction in tissue morphogenesis, we have found that cell junction plays an important role in the process of tissue morphogenesis. They are related to each other and can't be separated. Cell junctions are present mostly in epithelial tissues and responsible for the interaction of cells and shape forming. It is proven by a research where the physical basis of those interactions became clear when tested by electron micrograph. In tissue morphogenesis, coordinated changes of the shape of the cells are very important. Morphogenesis will occur when the mechanical properties of the cell change, such as when the cell divides, apoptosis, or forced by the environment into the epithelium. Therefore, the rearrangement and changes of shape greatly influence the process of tissue morphogenesis, and the presence of cell junction is needed. Besides the importance of cell junction in tissue morphogenesis, some disease disorders can occur because of some dysfunctions or abnormalities in cell junction. Those disorders can lead to various pathological conditions. So, treatment is the only way to treat those disease disorders so that the process of tissue morphogenesis will not be obliterated. That's all from me. Thank you.
Good morning, my name is Nyoman Putra, my last student number is 050. I will tell about the conclusion of the discussion before. First, cell junction are specialized area of the plasma membrane that attach one cell to other or to extracellular material. Second, tissue morphogenesis is the process of tissue development and differentiation that occurs as a result of cell junction. Third, dysfunction result in various pathological conditions, and the treatment for disease disorder of cell junction in the tissue morphogenesis is different for each adhesion system. Next, there's also some recommendation that we have, which first is the vital thing is to be studied by the pharmaceutical and medical student because cells are the smallest units of life. Second, by understanding how cell works in the medical world, modern drugs that are more viable can be treated with superior quality. Hopefully, this can be used for reference in studying cell biology for pharmaceutical and medical students. Third, this research is also very valuable to us, the writers, in understanding more broadly in the lecture material that we study in this lecture. That's from me. Thank you.
That's all for our student project presentation today. We are sorry if there are some mistakes during our presentation. We hope that our student project can inspire and educate a lot of people based on its benefits. Lastly, thank you for listening and watching and giving your attention. And I would like to close the presentation with the Paramasanti. Om Santi 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 Om. Thank you very much.